Dear students, welcome to Commissionerate of Collegiate Education. I am G.L.N. lecturer in Zoology, Government College, Autonomous, Anantapur. Today, we are going to learn about the basic properties of antigens. The learning objectives of today's session is, we are going to learn about the nature of antigens, followed by the size, followed by complexity, antigenic determinants, specificity, and classification. So we are going to throw light on these aspects of the antigens. We are going to learn about the nature, that means physical nature, chemical nature, and size, that means what is the minimum size for a molecule to act as antigen, followed by the complexity. And what are the antigenic determinants? There are certain structures that are present on the antigens that determine the immunogenicity of the antigen. So we are also going to learn about that. And specificity. Each antigen is specific. That means they exhibit some sort of specific nature. Followed by classification. How they are classified. That means there are certain classification criteria. Antigens are classified into exogenous, endogenous, like that. We are going to learn about these dimensions and dynamics. Now, coming to the definition, antigens are the molecules that are foreign to the body. So, antigens are the molecules which are not native to the body. These should be foreign to the body. And they should have the capacity to trigger immune response. That means all the foreign molecules can't be branded as antigens. For example, if you take some polythene paper, plastic paper and swallow it, you can't expect immunogenicity or antigenicity. That means any molecule that will trigger the immune response in your body can be called as an antigen. And here I mentioned that they trigger the generation of antibodies against them. That means the antigen is enough to trigger the manufacture or synthesis of antibodies. And these antigens and antibodies come together, they act in lock and key mechanism. As a result, the activity of the antigen will get nullified. Antigens that trigger the immune response are called immunogens. That means all the antigens may not be immunogens. That means all the antigens that enter into your body may not be in a position to trigger the immune response. Any antigen that trigger the immune response is called immunogen. Antigen is represented by the symbol Ag. So that is about the definition and the primary aspects of the antigens. Antigens are mostly proteins. No doubt, certain polysaccharides may also act as antigens. So proteins and peptides especially, they have got more antigenic nature when compared to polysaccharides. But polysaccharides coupled with proteins, they will again have that capacity to trigger the immune response. Whatever may be, these are the chemical factors that can act as antigens, namely proteins, peptides, or polysaccharides. But the capacity of polysaccharides to act as an antigen, that means the capacity of the polysaccharide to trigger the immune response is less when compared to the proteins. And the lipid and nucleic acids can also act as antigens when they are coupled or when they are mixed up or when they are combined with protein or polysaccharides. So that means previously we learned that proteins and peptides are better antigens when compared to polysaccharides. Along with the polysaccharides, even lipid molecules and nucleic acids can also act as antigens, provided they couple themselves with protein factors or polysaccharide factors. Minimum size of an antigen. This is the size, that means physical aspect of the antigen. The minimum size of the antigen is considered to be 5000 Daltons. That means if any molecule, if it is less than 5000 Daltons, it cannot act as an antigen. Low molecular size substances may also act as antigens, no doubt, but they should be combined with the large size molecules. That means if the molecule size is smaller and smaller, although it has got the capacity and properties of the antigen, it will be more effective if it is combined with a larger molecules. Haptens are the low molecular weight substances. You can give this haptens for representing partial antigens. I told that the low molecular size substances may act as antigens, no doubt, when they coupled with greater size molecules. Like that haptens, they represent low molecular weight substances. They act as partial or incomplete antigens. So this is the best example for partial or incomplete antigens. Now coming to the definition of immunogenicity and how it is determined. The immunogenicity of antigens is determined by the molecular size and complexity. These two are important. Smaller molecules may not be in a position to act as an antigen or effective antigen. Like that, complexity is also important. That is the reason why proteins act as better antigens when compared to carbohydrates. And carbohydrates will act as better antigens when compared to lipids. So low molecular weight substances or low molecular size substances are not that much effective antigens. So for example, I have given certain examples also in the previous slide. The peptide antigen, that means the protein antigen, may have a repeated sequence of a single amino acid or multiple amino acid. You know pretty well that protein is made up of amino acids. If any antigen, protein antigen, it may contain a single amino acid repeats or multiple amino acids may be repeated. In such case, you see this aspect, a molecule with 2 or more repeating amino acids is more immunogenic in nature. That means if you compare a molecule with a single amino acid repeat, if you compare this with a molecule with 2 or more repeating amino acid units as repetitive units, that means the molecule with 2 or more repeating amino acid units is more immunogenic in nature. That means greater the complexity, greater is the capacity of a molecule to act as an antigen. Now, antigenic determinants. What are these antigenic determinants? Every antigen will have a specific site on it. This site is called epitope. Like that, antibody will have one particular site that is called paratope. That means epitope is present on the antigen, paratope is present on the antibody. When they come together, the antigen and antibody, that means epitope that is present on the antigen and the paratope that is present on the antibody will combine together like a lock and key. Like that, epitope and paratope are acting like or else interacting like lock and key monuments or lock and key dimensions or lock and key dynamics. So this lock and key mechanism is very crucial in order to fix this paratope into epitope. This epitope is called antigenic determinant. So what are antigenic determinants? Antigenic determinants are known as epitopes. Epitopes are nothing but the sites that are present on the antigen that are going to fix up with the paratope. And if you compare this mechanism of epitope and paratope, they act like a lock and key mechanism. Okay. Epitopes are those specific regions on the antigens that bind with antibodies. These epitopes play a significant role in immune recognition and the response. That means epitopes are very crucial in triggering the immune response. Antigen may have multiple epitopes. There may be single epitope on the antigen and one antigen may contain multiple epitopes, multiple reacting sites on it. And there are two types of epitopes, namely linear epitopes and conformational epitopes. Linear epitopes contain a continuous sequence of amino acids or sugars. Coming to the conformational epitopes, these are formed by spatially distant parts of antigen, but they come together or they come very closer, they come very nearer when molecule attains a 3D form. When it is a normal form, these are spatially distant, but once the molecule attains the 3D form, they come together, they come closer with each other. And here you can see, I have listed out the different types of antigen. For example, microbial antigens, they include bacterial antigens because every bacteria will contain protein material, that means it is covered by glycoprotein structure. So this is enough to trigger the immune response. That means bacterial component, viral component, fungal components, parasitic components can act as antigens. So I have mentioned that antigen should be a foreign material. All these microbes, that means viral and bacterial, fungal elements, they are foreign to our body. They enter into our body, and because of the outer coat, outer covering, which is made up of the protein, they trigger the immune response. And you can give Plasmodium as the best example for this parasitic antigen. And the beauty of Plasmodium is it always changes its outer protein coat. As a result, your immune system doesn't recognize this Plasmodium antigen. This is the reason why Plasmodium is called a smart parasite. It is a smart antigen, it contains smart antigens, it is a smart parasite. Even HIV virus is also a smart parasite. So coming to self-antigens. Normally self-antigens are present within your body, within the body of an organism. For example, you take the lens. Lens is made up of a protein. And I told that protein can act as an antigen, but why lens is not triggering immune response? The reason is lens protein or lens normally, it doesn't come in contact with blood circulation. If any protein comes in contact with the blood circulation, then only it will be recognized by the immune system. So normally your lens that is present in the eye, it's not exposed to the blood circulation. But due to injuries, if it is exposed to the blood circulation for the first time, then your immune system will recognize it as an antigen and it starts producing antibodies against your lens. As a result, your lens gets spoiled. And my dear friends, sometimes even the antibodies will be generated against the spermatozoa of the same individual. If the spermatozoa get mixed up, that means if they enter into the blood circulation due to some reasons, due to some injuries, when the spermatozoa are released into the vagina during the process of coitus, if they enter into the blood circulation, then what will happen? Antibodies may be produced against the spermatozoa within the female sometimes, that means in the partner, or else sometimes if there is injury in the genitalia, male genitalia, and if the spermatozoa are exposed to the blood circulation of that particular individual, even in such cases also, the antibodies will be synthesized against the own spermatozoa. And allergens, these are very common antigens. If you go to Bangalore, which is nothing but the garden city of India, you know pretty well that the pollen grains are just suspended in the air. And if you inhale this, it may cause some sort of allergies. Any antigens that cause allergic reactions can be termed as allergens. Coming to vaccine antigens, and it's very common. Nowadays we are taking vaccines which are nothing but attenuated viral particles or where the disease causing capacity is totally reduced. That means weakened microbes. Weakened microbes can act as vaccine antigens. And you may be knowing this blood group antigens. You might have studied it in genetics also. You know that we belong to 4 categories of blood groups, namely A, B, AB and O. And this entire blood group classification is based on the presence of antigens. If B blood group is given to the person who is having A blood group, it results in incompatibility. A antigen and antibodies, that means the antigen that is present in a particular individual and the antibody which is incompatible that is present in another individual, they agglutinate with each other. That means they such reactions, such antibody antigen reactions may result in agglutination here. In some other cases, it may result in precipitation. So whatever may be, there are toxic antigens also. There may be tetanus toxoid. You might be knowing one disease called tetanus. That tetanus toxoid is a toxic antigen. There may be tumor antigens also. We will learn it later. Structure of antigen depends on nature, size and immunogenicity. Here you can see the beautiful pictures of antigens, and the number of epitopes may vary with different antigens. I also already made it very clear. And there is one more classification regarding the antigens. Probably this may be the last slide or penultimate slide. And exogenous antigens and endogenous antigens. The name itself indicates exogenous antigens are from outside the body, whereas endogenous antigens are originated within the body. Exogenous antigens will get access into your body either by inhalation or by injection or by ingestion. Whereas the cells within the body, compounds that are prepared in the metabolic activities and fragments of the body, they act as endogenous antigens. Apart from that, there are tumor antigens. You can see the tumor antigens here, cancerous antigens. So this is about the antigen nature and antigen classification, definition of antigen, my dear students. And at this juncture, at the outset, I should be thankful to CCE AP Vijayawada and my studio supporters, Dr. Chandrasekhar and JKC coordinator Swami. Thank you very much.